And now we will proceed with the session AI and Big Data. We have with us Professor Song Si-young of Yonsei University, who is going to be the moderator. He's going to lead us into the changes in the industry with the use of technologies.
It
is great to be here. Welcome everyone to Bioplus-Interphex Korea 2021. This conference is sponsored by Samsung Bioepis, Hanmi Pharma, Envirotainer, Macrozen, Sartorius, and 10 other companies. And I will be the moderator for this session, AI and Big Data, Biohealthcare Innovations New World. I am Song Si-young from Yonsei University. We have 5 presenters with us, and these are the foremost experts in these fields in Korea. I believe that there have been more than 2,000 people that have signed up to listen to today's session, and I think that we can feel the great amount of interest in this field, and I believe that this is indeed a direction that bio-big data is taking. We have bio-big data, hospital data, and gene data in bio-big data, and in Korea we have the different acts that were revised for big data, but we still have hurdles in many fields. And from this perspective, in March of this year, Deloitte Insight covered a questionnaire for the CEOs of major pharma companies in the US about what are you preparing for 5 years going forward, what is your utmost priority? And they have answered curative drug development has the highest degree of completeness, but what needs to be more invested in is in digital and security. So that is AI and big data. In Korea, we have the medical field and we have R&D for health, and I believe that compared to other countries, we have highest potential because we have a huge amount of support by the scientists and the government. And I believe that our potential is about to bloom in digital for AI and big data. The hospital data is very important, and many hospitals have invested hugely in this field to lead to convergence. However, there are many hurdles that we need to overcome. We need to have convergence between the hospital data between different hospitals, which is difficult. And we need to converge different data from medicine, biology, and other fields, but that is difficult. So let us look at what is the current status of AI and big data and hear from the foremost experts in these fields. We have National Bio Big Data and Lifelong Data Integrated Data Dam project that is being pursued. So coupled with these projects, we have the companies, universities, and many people that are involved. So let us try to think of the future.
We have as the first speaker Dr. Kim Jae-hak, who is the CEO of Alan. He has graduated from the School of Pharmacy at Seoul National University. He has worked in CJ Boryong Pharma, and CMG was what he headed, and he has developed cytopsin for high blood pressure, and he has led such outstanding projects. He's going to talk about the new drug discovery strategy in the age of AI. Please. Hello, everyone. I am Jae-hak Kim, the CEO of Alon. I'd like to talk about the new drug discovery strategy in the age of AI. As was just introduced, what we have now the advent of the age of AI-driven drug discovery. So I'd like to talk about the short-term and long-term strategies for AI-driven drug discovery and wrap up with the future of AI-driven drug discovery. It's been about 10 years that investment has been made into AI drug discovery. And this year, the investment is expected to reach $4.5 billion. And we have the rankings, as you can see from the graph, UK, China, Singapore, Hong Kong, Canada, and France. And we are missing Germany and Japan, which are the usual strong players. And as for the R&D, in 2009, it was we see that the graph is falling exponentially, and the clinical study cost is rising, and the number of drugs approved per $1 billion used to be less than one since 2009. Cost per one FDA-approved drug is $2.6 billion. Time to be approved longer than 10 years. And clinical trial success rate less than 12%. And AI and big data is the solution to this problem. And this is the DSP for 1181, developed by Excentia and Sumitomo, and in HC Comedy used the AI and was able to select the target and was able to find the hit compound in 46 days. And this was published in Nature Biotechnology in 2019. And in silico medicine used $2 million and was able to come up with hypothesis to preclinical drug candidate in just 18 months. And as for Google's DeepMind, it unveiled a 3D structure prediction technology, AlphaFold 2, and databases. And this made it easier to discover new drugs using AI technologies. And even though we have the AI technologies, the collaboration of AI technology and wet labs is still the efficient approach. AI and wet labs two-track approach was able to reduce the cost by 1/3, and AI new drug development has some issues because synergistic computational model, quality DB, it's difficult to standardize for testing and validating the results. And it has issues with multi-domain proteins, and there needs to be procedures for multi-drug effect assessment. So we need to take caution and use AI technology partially. And we are able to identify the CAD using image domain, but in the chemical domain, the phenotype, dose, endotype could introduce some variability. And as for the biological domain in drug discovery, depending on genotype, dose, it is heavily conditional. So it would be difficult to use AI for new drug discovery. And so because of the high variability in biology, what we can do is we need to focus on the chemistry area, because it is easier to solve issues in chemistry using AI. And because the underlying principles of thermodynamics it can determine the affinity of a ligand for a receptor are well defined. But for complex biology, equilibria and biosignaling is much more difficult to understand. Let's say, for example, the structures and values can be given to help augment chemists' thinking. And we have the methyl ether, and it is changed to sulfone. And on the left-hand side, on the bottom, it is changed to acetal. And log-deal is changed to 5.1, and PIC 50 is 0.9 improved. And second example, This is a heterocycle and methyl and chloral. And in the middle with the media, carbon space is added. PE50 solve and HERG is 5 times, but the traditional method would take about 5 years, but using AI, you can shorten the cycle time to just 1 year. And as for the long-term strategy, it's a matter of whether you want to develop a drug or a ligand. Integrating both ligand protein activity and target identification along with the PK properties of the compound in an integrated manner is still missing when it comes to AI in drug discovery. Understanding the certain efficacy and safety related processes still need to be improved to base practical decisions on such models, and you have a higher chance of just remaining at the ligand development stage. So you need to take it up one notch, look at the signal pathways, and multi-type data needs to be used, drug design, and other data and information needs to be looked into very carefully. And talking about the future of AI drug discovery, the pharmas they're increasing in collaboration amongst one another and also with IT companies. And within 5 years, drug discovery using AI will experience a quantum jump. And AI and drug discovery technologies. So AI companies, Essentia, Flexon, Lantern, Tarbin, and Schrödinger, and there are Diazen as a Korean player. And now DT, digital transformation age is upon us. Nokia and Kodak did not survive. Microsoft, Google, Amazon was able to adapt to the changes and was able to grow. And the core of AI-driven and data-driven new drug discovery is innovating the ecosystem. And if the pharmas take this to heart, they can also survive from the competition. The AI model is taking place in all the processes, research and target identification, generation of molecules and predictors, and can save cost and time. And we have many global pharmaceutical companies working together on the Melody project for database, and the need to work together to build competitiveness in AI technology will grow. The take-home messages are the possibilities of drug discovery using AI is infinite, but we need to look at the advantages and disadvantages, and we need to understand the issues. And as a short-term strategy, it's necessary to focus on the physical-chemical data, which is relatively clean, and cooperate with Medichem team rather than focus on relatively complex and variable biology data. After all, in order to develop a drug and not a ligand, it's necessary to consider deeply what biological data can be used. With this, I'd like to conclude. Thank you.
감사합니다.
Thank you very much. That was a very compact presentation. You were able to show us the short-term and mid to long-term strategies for new drug discovery using AI. Thank you for the enlightening lecture.
Let us go to the second presentation. The second presentation will be delivered by CEO of Standime. CEO of Standime, Kim Jin-han, is a person who graduated from Applied Chemical Department and also received a master's in computer engineering and has worked for NC Software and Samsung Institute as well. And Standime is currently a great company doing research in new pharma development related AI. I would like to ask CEO Kim Jin-han of Standime to deliver his presentation. Greetings. I am Kim Jin-han, the CEO of Standime. I have been included in the AI and big data track in Bioplus-Interphex Korea 2021. And I will be delivering a presentation titled Artificial Intelligence Drug Discovery Technological Strategy on Best-in-Class to First Class. How can we transition? This is my table of contents. First, part 1 is when there is target protein and there are new ligands that could generate a molecule. So I will cover that part. Part 2 is novel target discovery. And this is not to discover proteins for the first time, but for the ones that have not been targeted well yet or that we lack knowledge about. These refer to targets where the protein has a possibility to be linked to drugs. Part 3 will be bridging between the novel targets that we may have created and the novel molecules that we would like to create. And there are some cases when there isn't enough information about the target structures and how we can create and design ligands during those times. What I would like to talk about is not a talk centered on my company, Standime. So it will not be focusing too much on the business aspect, but I will be talking in more of a general academic sense. So I will be speaking under the premise that this is a more academic and general talk. The slide that you are looking at is regarding drug discovery and development. And you probably are very well aware that we have target discovery to lead the animal models clinical trials to FDA approval. So this is comprised of these series of activities. Of course, we can see that as a premise in the clinical phase, there are many areas where AI could be used, and there are other areas such as patient data that exist. There is the genders of the patients and others, but there are some areas that Standime is focusing more on since we have more data and since we believe then can lead to a higher ROA, which is the first area of target discovery. And secondly, it is the area to lead, which is about creating the ligand. So this is until the stage when it can then go to preclinical. So this is the area that we are focusing on. Part 1 is what I would like to talk about now. This refers to technology to create and optimize the ligands for the given target. In the community that is involved in AI drug discovery, this is called de novo molecular design and generative model. And the first is to create a landscape that did mechanical learning or deep learning. So creating the landscape is the beginning. There could be other ways, but regardless of what they are, is that with chemical data, learning takes place, and the result of the learning is taken to make abstract patterns. So to extract the abstract abstract patterns. So this is a scape comprised of those patterns. This is also called chemical space in short. And this can be divided more thinly according to the technology, but you can see molecule database like zinc that has bigger numbers. Learning is done for those. And regarding who is the subject of learning, in the case of medications or drugs, then the subject of the learning is that there are not many cases when there are the properties or names on the labels, and most of the materials just exist as what they are. So we need technology so that the properties of the material can stand out. That is, to see what is the innate structural characteristic or property of the molecule and what kind of properties can congregate. So this is a panel where the structural characteristics that were learned through heightened abstraction can be expressed. This is the landscape, and some are known by researchers and the community, so we can put a label for those. And you can see the labels that have good properties are in red, and those that are not known or not good are in blue. Thus, based on the learning structure when generating material, we will distribute this on the landscape, spray it on the landscape, and in the landscape, when something is near the good points that we already know about or along the landscape gradient, it can move to a better point following the gradient. And if the inference is that it can be a better material somewhere in between, then that is when we do a sort of decoding. Then a new structure will appear, and when that happens, we can extract new material at a better point. This kind of molecule is called a representation. And when this is used in machine learning, deep learning, how are we going to make it learn and express, and what kind of unit are we going to divide for this to learn? That is the area that we are also involved in. And when there is a material, a molecular structure, we need to synthesize. So the basis of structure reaction, the reactant exists. So the structure is divided in those units, and representation is formed and then learned. That form exists. And there's also MD, which is more focused on energy calculation or charge distribution. So learning is based on this, and structure is created, which is called atom-based. And there's another method when this is divided into scaffold units or functional group itself or into fragments and then learned. So there are largely these three categories. In our case, according to the goal and the stage, many expressions are mixed, learned, and used. This refers to how the structures can be learned, how new structures can be created, or how the new structures can be found, and what kind of method can be used. It can also be largely divided into gradient-free and gradient-based. Gradient-free makes the many chemicals here bind and hop, and other chemical structures are navigated and created. So this has a discrete form. Maintaining an already good structure is a capability, but to go over the structure and navigate a completely new structure is another benefit. Next is gradient-based, and this is a form used often in the deep learning category. And This is to make the chemical landscape that I showed you beforehand and decode following the landscape gradient. A case in point of the gradient-based deep learning field is the technology variant autoencoder. When there is the autoencoder, basically, when there is a molecule, in order to get the characteristics from the structure, there are the layers and your own structure then pop out. And it is to get a layer with a high degree of abstraction. That is the autoencoder. Variation autoencoder is a basic autoencoder. The material does not appear, then based on the existing material as a seed, the goal is to get another material with better structure. So that is the goal, and that is why we say that there is a variation. This is a type of sampling, and the variation is to get good sampling from the distribution. When there are materials which is better or different in structural from the existing materials, you can see what happens. And another technology that Standime is applying is point cloud. This is a higher level than the representation that I aforementioned, and it will reflect 3D structure on the materials. And the 3D structure is used often in fields such as image recognition in points. And this is to apply this to the material space so that the 3D space structures can be captured in a much better way. And this is a research method for that. Standime Best demo exists, and I would like to show this to you. And I have played the demo. You can know how it proceeds in this way. We start from the target DB, and there is an area when we choose the target, and you can choose the already known areas from the compounds from the target and DB. And when you choose the target, on the virtual screening, there's the PharmaCore model, which is applied, and good material that can act as the seed can be selected. And this is that kind of information. Detailed information can be seen in the workflow by Standime, and this is an area making a new scaffold. This is using a model called Monet to keep on generating derivatives and finding good derivatives. The whole project can be monitored and managed in this manner. This is a basis for us to maintain a pipeline with productivity. In addition, Standime, although there are many other companies in this field, we are included in the leading group, and we have the basis to make novel targets and novel materials. And we're not only making a platform, but through collaboration, we can directly implement pipelines ourselves, and we are making new materials ourselves. And early next year, within Q1 of next year, our timeline exists for preclinical candidates. Excitia, the leader, is in phase 1, and in silico medicine is in this stage, and we are about next in the progress. The next target is novel target discovery. Standime-esque, the open version iClone-esque, as you can see on the screen, exists, and iClone-esque is an open platform, and you can join and log in and use this. It is to let a target and disease queries to be known, and to get the potential targets for that disease, extract any proteins that can be similar. And these are points and standards, and you can analyze them through the graphs. This is a type of platform, and it can be divided largely into three. There is the GraphDB, which is comprised of nodes and links, known papers, curated DBs that can be taken real time, and all of this is integrated so the nodes, whether it's a disease or protein or pathway side effects, you can see the data is for that, and the link is the relationship. Algorithm is when there is a disease given, the specific patterns in the specific pathway level, which exists in between, which can extract targets. So having the pattern for different materials on a meta level, this is for the researcher to interactively set a hypothesis and then go through this. This may look small, but these are the representative databases that we are integrating, and this is explained in the iClonist basic version. And using the latest BERT model, you can see in a paper getting real-time nodes and edges, and we are integrating them. As I mentioned, the diverse databases are comprised of diverse edges and nodes, and as I aforementioned, the algorithm between the disease and the targets, there will be many pathways and the generalized pattern, and the relationship between the disease and the target, which can treat it. And this is made into materials, so it is learned. So when a new path is proposed, it is to see how good the path looks and to actually give forth a hypothesis. This is also a demo, and this is on YouTube. When you log into iClonesc, you can see how it moves. You can put it on query and to see how the nodes are extracted and how the relationships are created. You can navigate. You can also see the interpretable evidence come out of the papers and new materials. For the 4 diseases, 4 indications from IPF to Parkinson's disease, we set new targets and predicted and saw the experiment results, and through collaboration, and we also did it by ourselves. And for IPF, there were 3 targets that we can explain to you, and they went through the test and validation, and we have secured the primary level targets, and we will engage in more detailed mechanism validation tests going forward, and all others are in the validation test phase as well. Now I would like to cover part 3. If we can design and optimize materials, and in between these technologies and the technologies to uncover novel targets, what kind of issues can exist and how can we overcome these issues? Basically, when we say that we found a target for a disease and we want to make a molecule for that novel target, that target may have a well-known crystal structure. And when there is a structure, even if we do this with AI, generally, since there is a target structure, ligand design can be done, or we can have the approach in a more detailed way, generally. Secondly, is when there is insufficient information and the structure is not known. As you probably know, through homology modeling, the structure is created to the utmost of capability in the case of the unknown structure. Next is AlphaFold method. This is a case in point. When there is only the protein sequence given, it is to make a 3D structure. And with this 3D structure, it is to design a ligand. For the AlphaFold, it has quite positive results. And nowadays, you can see that it is meaningful as a solver. Of course, it was overwhelmingly number one in the challenge, but it is also meaningful as a solver because the results of the experiment and the creation of the 3D structure using an AI model did not show much of a difference. So it was nearly equivalent. You can see for AlphaFold 2, those numbers are shown, and that is why, regardless of whether you do the test or not, and whether we make a prediction with AlphaFold 2, there were predictive results. Standime is also developing this, and when there is something with an unknown target, when that is given, then for some exemplars, the sample ligands are utilized well, and we engage in first round of tests. And from the sample, we can predict the target structure inversely, or we can use the common characteristics of the collected ligands as seeds to go over the design. And we are currently developing this method as well. We are using many methods to approach this field so that we can fill in the gap between the novel target and the new compounds. These are the technologies from novel targets to compounds, and we call this internally a series of workflow AI. And these are some new projects when we use these series of systems. We have reached hit to lead stage, and these are all for oncology diseases. You can see that they were done according to the numbers of workflows. So you can see stage 1, 2, and 3. These are the results, and you can see results of 3 and 4 that are forthcoming as well. This is the overall picture, and within 7 months, we wish to get a lead equivalent material and then go forward and lead the way. That ends my presentation. And please contact me if you have any questions. Thank you very much for listening. Thank you very much, CEO Kim Jae-han. It was a very detailed, and you gave us a very good overall direction. In Korea, we had to focus on best in class, but through this new technology, I believe that we can have new materials for first in class, and I hope that we can become a leader in bio. A point of concern is regarding the AlphaFold that you aforementioned in the Google competition that was introduced, and they made a pharma company this year, and they might use that platform to collect all the data from the world. And there will be YouTube and other ways that they will be utilizing this data. So we need to think about how we can get synergy by converging our data as well. We heard about new pharma development related AI from the two presenters.
Moving on to the third speaker, he's going to talk about the new era of digital transformation in healthcare. The speaker is Dr. Hae-sung Lee. He is with the KT Future Value Office, Digital and Biohealth P Task Force. He has graduated from Sungkyunkwan University and has completed his doctorate degree in biomedical from Sungkyunkwan and Illinois Institute of Technology, and also biomolecular chemistry from Pohang in MS. Dr. Lee, you may begin your lecture. I think there is a bit of a latency. Do we have the microphone connection on? Dr. Lee, we cannot hear you at the moment. Could you check your microphone?
시작하도록 하겠습니다.
Let me begin. Yes, please go ahead. Hello, everyone. I am Haesung Lee at KT's Digital and Biohealth P Task Force. I'd like to talk about the digital transformation taking place in healthcare. Let me begin again. I apologize, there were some technical issues. This is the two-part presentation. I'm going to talk about the digital transformation in the medical area, and then secondly, digital transformation in the pharmaceutical area. First of all, in the medical DX, the earlier speakers mentioned this a lot: digital therapeutics, telemedicine, medical AI solutions, digital surgeries, and personalized health. I'll touch upon these very briefly. So I think this is known to most of you. The digital healthcare is now moving away from cure focus to care focus, which is more preventative. So it's decentralized and personalized. The healthcare is becoming more diversified. And as of now, looking at the trends, as you're well aware, we can compare it for before and after COVID-19, telemedicine, mobile healthcare, and wellness. We see that the market is growing, and in the process, we see a lot of the digital transformations taking place. And healthcare business is becoming more diversified and is being upgraded. And the boundaries in the businesses are coming down. And so we have these disruptive innovations. Amazon is delivering medication, and Vuno and Lunit is also undergoing changes in Korea. And so this, again, is quite familiar. We have medical data and genomics data and life log data, and they lead to P for medicine. The P's stand for preventative, predictive, personalized, and participatory. And this is the future trend of where the medicine will go. And to go into a bit more detail, as for the health data, they used to sit in disparate systems, but by building the My Healthway platform, we'll be able to integrate health data, hospital medical data, and the public health data, and they will be integrated, and we'll have single access to this, and this will create a new opportunity for a new market. And the health data amounts to thousands of terabyte data, and ICT technology encompassing AI, big data, and cloud. And if they merge with financial data and insurance data, then this will be able to serve as a foundation to provide meaningful services to the consumers as well as the providers. So the sum of the trends is digital therapeutics, telemedicine, medical AI, digital surgeries, and personalized health. And these are the areas where medical transformation, digital transformation will take place. Let's first look at the digital therapeutics. When we talk about digital therapeutics, they will play diverse roles. And this will fulfill some of the medical needs that were not satisfied before. Many different options will be provided to the users. and it could be used in companion with the other therapies to make it more effective. And it could be used to improve the existing therapies or to reduce the reliance on certain therapies or certain medicines. And we call this SAMD, software as medical device. And looking at the market size, as of now, looking at the advanced nations, they are leading the growth, but Europe, the North American markets, Japan and China are following suit. And we see that the CAGR is around 26%. And looking at the areas, management of chronic diseases such as diabetes, obesity, depression, and addictions, and behavioral changes, how about quitting smoking and making the right consumption choices, and medication compliance, and data management. And I think you've seen these examples. This is Ikeli in the game format and pair therapeutics to reduce the drug addiction. And NeuroSigma is using patches to stimulate the brain waves to treat ADHD for children. And for new NAPs, they are using the VR device as a software program to rehabilitate the brain damage from stroke. And I'm sure you've seen this paradigm. The benefit of telemedicine is you could have access to medical services anytime, anywhere, and action can be taken. And the mothers, when they take their babies to hospitals, physically, they had to wait a long time and they had to make appointments way ahead. But you don't need that much in telemedicine, and there could be collaboration between different doctors, and you could have continuity in the treatment. So this is a very promising area. I know this sounds like a textbook, but you could have more collaboration and convergence with the ICT technology. Of course, the regulations are changing with the COVID-19 pandemic as a deflection point. And so telemedicine has been allowed temporarily due to the pandemic, and we will be monitoring the situation in the digital industry and in the telecommunication industry. We see the benefits of telemedicine. And the CAGR for the telemedicine is 25%, and we can divide the telemedicine area into 3 markets. The first sub-segment is hardware, the second is medical services, and the third segment is software. And we see that the hardware plus medical services add up a lion's share of the telemedicine market. And we see the strong players of the US, Japan, the UK, and China. And we see that the markets are growing in different segments. And as for the medical AI, AI is undergoing a lot of changes and is being used in the areas of prediction, diagnostics, and treatments, but mostly they are used for predictions using the imaging data. And as for sepsis or arrhythmia, AI could be used to predict these diseases. And ultimately, what we are targeting is we want to. There was an investment made in Lunit, and AI new drug discovery platform could be used and could be used as companion diagnostics. And another area of digital surgery. We are seeing digital transformation, and so pre-op, during the operation, and post-op, we could have rehearsals before the operation using the 3D imaging so that we can optimize the operation setting, and we could reduce the pain, and we could optimize the operation setting. And during the operation, we could have minimal invasion, And we could have AI or AR used. At post-op, we could predict the outcome of the surgery. And this could also be used as a solution to supplement rehab. And these are values to be added. And we have Korean companies, TNR Biofab, 3D printing company, and Hutom. AI-based imaging analysis platform is being developed. And as for Neofect, it is a startup that provides rehab solutions. And so, as I mentioned earlier, they were just brief glimpses, but diagnostics and predictions are used for personalized health. And in oncology, we could have fast treatment. And as for cardiovascular, we need some more time. But liquid biopsy and companion diagnostics are seeing some results. So we could see continuous medical digital transformation take place. AI technology, biosensor, digital pathology using AI solution, these could be applied to personalized health, and data-based and imaging-based diagnostics are using AI solutions. And we could have more biosensors and diagnostics devices coming online on the market. And now let's look at the digital transformation in pharmaceutical. Other speakers talked about the AI drug discovery, so I'll be very brief on that, and I'd like to talk about the clinical trials. So the overall trend is in a new drug discovery, the complex processes could be streamlined through digital transformation. You could have AI drug discovery, and you could have virtual clinical trials, and logistics and marketing could be digitized. And pharmacovigilance could be done based on data, and this allows for many new opportunities in digital transformation. So for AI drug discovery, as you're well aware, there has been a paradigm shift because they used to focus on the symptoms of diseases, and now they focus more on the omics and the knowledge, and they want to intervene at the mechanism of the disease, and this can save a lot of time and cost and minimize the risks, so that data-driven monitoring is possible. And so the development process and the post-management processes can be done more effectively. And this is just a slide for examples. We have global pharma companies and biotechs and IT companies and GPU and data processing companies are collaborating under open collaboration platform. So target discovery, and in clinical settings, there's a lot of collaboration and innovation taking place, and M&As and equity investment is also done to come up with quick results. So there's a lot of innovation going on. And looking at the clinical trials, it used to consume a lot of resources, and it used to be done through CRO, face-to-face. But we could now have a hybrid. Data collection is done, and sometimes some of the work is done virtually. And so we have the DCT, decentralized clinical trial, or we have the hybrid clinical trials. And because and looking at Moderna's development of the therapeutics for COVID-19, smart devices and regional and community clinics could be used virtually. And regulation-wise, in Korea, this model is not used much. In Korea, the clinical trials and their documentations are becoming more electronic and digital, and patient reporting and data analytics are continuing to accelerate digital transformation, and we have the 5G infrastructure, and we have the security requirements. There's going to be convergence and drug prescription. The patients who are prescribed drugs, their vitals and their conditions will be constantly monitored through smartphones and wearable devices, and these devices will be used more often. And this is an example of a company, Science37, depression and lupus and Sjogren syndrome, restless leg syndrome. They are recruiting subjects for online clinical trials, and during pandemic, we had continuous clinical trials. Through the smartphone, the data was collected. So we will probably have more virtual clinical trials, not only in Korea. Telemedicine and drug delivery, if they're integrated, then we will have diverse formats of virtual clinical trials. And now moving on to PV. Pharmacovigilance market is expected to grow to a huge market in 2028, amounting to $14.9 billion, recording CAGR of 11.5%. PV is about getting the knowledge on adverse events of drugs and how to automate the PV using ICT technologies. And this is expected to reduce time and cost and to enhance productivity and efficiency. And this is one of the benefits that can be gained from digital transformation. I talked about the devices and pharma. So all in all, digital transformation is to benefit all, to provide equitable and advanced services to the consumers. And in order to do that, we need fast, accurate diagnostics, and based on that, personalized precision medicine should be provided. And we need remote management, monitoring, and rehabilitation. Then we could provide medical services and healthcare at a very rational cost, and we could have a very good health economy with this. I'd like to conclude. Thank you very much. Thank you. You talked about the digital transformation in healthcare. You gave us a very high-level overview of the changes. First in class is in drug delivery is for the global market, but digital transformation, I think you will have to target the domestic market first. Of course, there are regulations, but of course, we need to select the right market using the science and technologies, but we need to look at the demands and the needs of the people, and we need more promotional activities, and we need to have more quick wins, and we need to select our priority areas. And there are many interests involved, but telemedicine is a worldwide trend. I know there are conflicting interests, but there are many companies and researchers interested in telemedicine, and we need to resolve some challenges quickly so that we can go ahead with innovation. So thank you very much, Dr. Haesung Lee.
다음 강의로 넘어.
Let's go to the next presentation. The next presentation will be about EMR-driven big data, what happens in Korea. We will hear from Jo In-san, Brian Jo, the CEO of AvidNet. He graduated from Seoul National University and received his degree from Chunghwa University Medical School, and also from Elegant Korea and Hanmi Pharma. He has ample experience in new projects and currently is the head of a healthcare data company. He has more than 60 employees at his company and he is making a huge effort in this field. Please. It is great to be here. My name is Jo In-san, or Brian Joe, as was introduced. I will be talking about the EMR-driven big data in Korea and what happens in Korea. Today became very cold and many people have on heavy coats. You can see people wearing heavy coats and you can see that after wearing a mask for 1 year and a half, looking at people's faces without masks seemed to be a bit strange. So I think wearing masks have become a new normal. With non-face-to-face everyday activities that have started, you can see at a consulting firm in the US, they have shown us this data. This is about adoption of digital channels, which is a rising trend in all industries. The world seems to be getting used to digital and non-face-to-face. When we talk about pharma, we can't leave out clinical trials because in order for drugs and devices to be approved, you need to get clinical trials done. And you can see virtual clinical trials or decentralized clinical trials, remote data collection. Those have become buzzwords in clinical trials. There are diverse clinical trial patient-reported outcomes or the electronic consent, which are being done by smartphone. And I think they are becoming more and more widely used. You can see this picture, which was reported in the summer of this year. And I captured this because it was very interesting to me. And what this means is that in the US, in 2016, they passed the New Therapeutics Act, and they are pushing for real-world data. And they are encouraging this. What this means is that for the medical practices of the past, there is the medical data that came from claims data, and there are moves in the US by recent years about how we can utilize this data in more of a real-world situation. Regarding the approval of COVID-19 drugs, it could have become more challenging, but you can see the former US president and FDA former director who have come out and said that in order for the fast development of COVID-19 drugs, we need to accelerate real-world evidence efforts. So this was quite impressive to me, so that is why I put in this picture. These movements and using data to approve of these drugs and devices, I think is very relevant to precision medicine. Precision medicine includes huge amounts of patient data, and there is a great amount of analysis, and it is to provide customized data to the patient. In order to do so, we need big data, we need the my data of the patient, and we need ecosystem with good flowing data. So the US is emphasizing real-world data. So I believe that in the non-face-to-face situation, precision medicine is in a good environment to be accelerated. Going into real-world data in more detail, this is not controlled data like in the clinical trials, but these are EMR or registry claim patient-generated data that all falls within the realm of real-world data in the US FDA in 2017, 2019, and only 2 months ago they gave us industry guideline, so that it seems that they are actively using real-world data for approvals. If this happens in the past, there was limited usage of real-world data, but going forward, it seems that there will be more active use of real-world data. Last year, one company has said that among the companies that submit approvals to FDA, 60 to 70% have used the real-world data, and the percentage is going up. So you can see documents using real-world data and real-world evidence for FDA drugs and biologics is rising, adding new indications or label changes or research about safety, and others will be heavily using real-world data. We also need to focus on FDA and NHI, those nationally run institutions, are collaborating very closely with hospitals and healthcare companies. You can see when you search on Google, there are several titles that I have captured. FDA with the US health tech term is collecting real-world data for COVID-19. And there is Flatiron, another company that they are collaborating with for oncology. So you can see these activities are taking place. Let's look at an example. The US FDA is working with Asian, a AI company, and there are the randomized clinical trials that were done in the past, and they are trying to have duplicate studies to see whether this can be replicated in real world life. The results were validated so that the real world data can validate RCTs or that they can be utilized as evidence. So that was already done. I think we're living in a world where data is getting more and more importance. And let's look at what's happening to US big data companies. In first half of this year, at Google, they launched a product called Care Studio. Regardless of what hospital a patient visits, this allows a doctor to treat the patient. And I think this is being done as a pilot in 6 hospitals. So if you input the patient name, the patient's electronic health records can be seen. And Google has said that health data was very closed in the past, but they are trying to make this more patient-centric. It seems that we are going to soon live in this type of world, and this is similar for Apple as well. You can see PHR, personal health records, are now being inputted into iPhones. So iPhones can store patient records, and the patients can utilize this information when you go to a hospital. This is supported from iOS 15, and for iPhone users, if you update to iOS 15, I'm sure that you can get this support. This is not supported in Korea yet. We see these movements in healthcare tech and big tech, and the US government provided a great deal of support. There is a national health related institution in the US, and they had put in a huge amount of budget for the past 10 years so that they have made preparations for the data to flow from these medical institutions. And soon, the e-data of the patient will soon go to the mobile phones of patients, and there will be $1 million fine if this movement is blocked. In the US, giving this mandate and to implement this is quite rare, but in order for the flow of medical data, I believe that the US is making a true effort because they want to utilize the medical data and to create a ecosystem. I think they are showing strong intentions. Japan, that we have thought of as very conservative, is using more and more of big data. This is an article in the Korea Economic Daily that I captured. JMDC in Japan is a big data company in this field that has been growing very quickly. Even 5 years ago, they were quite small, but I think they have seen rapid development in Korea. There are efforts to utilize real-world data positively as well. And MFD has also published a book of real-world evidence and real-world data utilizing samples. And when we visit a hospital in Korea, the doctors access EMR, and many digital records are accumulated in the hospitals. There are many reasons behind why this data isn't well utilized, and this data is not pre-processed, so there is no standardization. We need to have pre-processing for the data to have significance when this is open. So from several years ago, there have been movements to standardize this type of data. As of November 2021, around the world, about 60 large hospitals have finished standardizing their data. You can see the medical big data stored in hospitals are ready for when the data becomes open. CDM, common data models, have been used to pre-process the data, and many national institutions, for example, HIRA or NHI, National Health Insurance Corporation of Korea, they also, I believe, are storing data with the CDM. What are the benefits of standardization of data? Of course, there could be some worries about the data going outside, but we can see the secure data in the hospitals that can be analyzed in a distributed form. So this is called a distributed data network, and Korea is currently setting this up for hospitals and for companies. If they ask for data to be analyzed, then the query on the data platform can be distributed to the entities that hold the data, and there could be a distributed data network that will only analyze the data that is relevant. A benefit of the distributed data network is that this is not just for one institution, but at multiple institutions, we can easily collect the data. This was a paper in Lancet in 2019. You can see that in a standardized form, they shared a query and they were able to analyze 5 million hypertension patients and to write a paper. This was something that was unimaginable in the past, but huge amounts of data can be analyzed in the times we're living in. We are also operating distributed data platform, and more than 1 million data queries are being processed in a distributed manner. So this is a very much active living data platform. The people that use these often are the medical researchers working at hospitals about the efficacy of drugs and about the efficacy of treatments are being analyzed with these types of platforms, and these are the people that are getting most significance. There could be many ways to utilize this data. When we engage in clinical trials in the different sites, we would have a feasibility for site selection, but with just one query, this can be done very rapidly. So in the selection and exclusion criteria for clinical data, it could be done very quickly, and making this a data platform could lead to quick confirmation about the numbers of the patients that are eligible. This is quite advanced, and this can be done very quickly. This type of solution is put on top of the distributed data network, and the user can directly process the data. Another way is AI training that could be done on this distributed data platform. The data is distributed, but using the federated learning method, on the different data servers, there are the models that have been trained that could be advanced. So global AI tuning could be done. And at my company, for diabetes, hypertension, and for high glucose levels, you can see for these diseases, we can make highly accurate predictions. So this platform is currently being developed. I have mentioned about predictive precision medicine, and I believe that going forward, based on big data, we can predict the next stage of the patient and assess the health status of the patient. So we are seeing many AI solutions. The government also is encouraging the utilization of this data. And last year in September, we had the pseudonym information processing guideline that was presented. So this is a very detailed guideline for utilizing these types of data. Another point of concern is for the individuals who own the data, to get consent from these individuals. We need to protect their data, and we also need to encourage utilization of the big data. So we need to have both. Using big data, for example, if we say we need to get patient consent for all, it would be quite challenging. The government, after revising the Personal Information Protection Act, has verified in the act some occasions when big data can be used without the consent. So there are some cases when the information is used for statistics or when it is for scientific research. Then in some cases, patient consent might not be needed. So I think that this is in a general form. This is a take-home message. In Korea, we have well-curated, rich, and valuable electronic medical data. We have overcome many standardization-related challenges. We have a guideline and a revised act for the medical data in Korea. So it seems that our environment for big data usage is blossoming. We have a good distributed CDM network, and this is seen to be very competitive in the global world. And going forward, although we do have most structured data forms, we will have more addition of unstructured data. And when that is added, then there will be a huge medical big data ecosystem that will be formed. Thank you for listening. Thank you very much for the insightful presentation. We have talked about many important themes of our times, and I am a doctor and I am a professor and I am in internal medicine. Many people say that in the 2030s, we probably might need internal medicine doctors who have knowledge in data. And it is said that more than 85% of what the doctors do in certain departments will be sitting in front of a computer. So thank you very much for your insightful remarks.
제목은
And the last presentation is predictive retrosynthesis, use and adoption by the chemists. We have two speakers. We have Dr. Abhinav Kumar. He is the Reaxis product development lead. And he has gotten his degree in pharmaceutical studies in King's College. And we have as the second speaker Dr. Moritz Klassen in 2012. He has started his studies in chemistry at the Ludwig Maximilians University in Munich and got his PhD in 2018. So let's listen to their talk.
Good morning, everyone. My name is Arnav Kumar from the Reaxys product team, and I'm here with my friend Moritz and collaborator Moritz from ETH Zurich. Today, we'll talk about Reaxys predictive retrosynthesis, its development, use, and adoption by chemists. Very briefly, I'll touch upon how the Reaxys predictive retrosynthesis tool was developed. Moritz will talk about its use and adoption by chemists in his research group, and we'll briefly look at the future roadmap. Small molecule discovery essentially needs to answer two questions. First, what to make, and second, important question is how to make it. And this is where we are focused on synthesis planning. Researchers and chemists spend a lot of time doing synthesis planning. So we asked the question, can data-driven approaches improve synthesis planning? This idea is not new. It has been around since the time of E.J. Corey, who won the Nobel Prize for having computer-aided synthesis planning. The critical requirements for any predictive retrosynthesis tool is one, it delivers scientifically robust predictions so that chemists can trust it. And second, it is user-friendly and intuitive so that chemists do not require to spend a lot of time in learning how to use the tool. The key elements for success of any predictive tool, not only retrosynthesis, but any predictive tool there are three key elements. One, the data science needs to be top-notch, i.e. the algorithm needs to be really good. This algorithm needs to be married with high-quality data to give robust outcome, to predict robust outcome, scientifically robust outcome. And third, that it should be delivered to users in a very user-friendly manner so that they can get on with it very quickly. Let's discuss algorithms and data. The success of predictive retrosynthesis models depend on both algorithm and quality of data that these algorithms are trained on. We believe that Reaxys reaction data offers the best data for predictive modeling. This is evident by the number of research groups and corporate organizations who use Reaxys reactions data to drive build new predictive models. Similar to the academic groups that have used Reaxys reactions data, we collaborated with Mark when he was at University of Münster, and Mark wrote one of the most cited works in the field of predictive retrosynthesis. His approach was a game changer because for the first time, deep neural networks and Monte Carlo tree search were applied to solve the problem of retrosynthesis. Through the application of these innovative technologies, the tool has learned to transform chemistry rules from Reaxys reactions data, has learned to prioritize those rules, and uses efficient search method to prevent combinatorial explosion. The tool is trained on 15 million unique single-step reactions from Reaxys, from which roughly 400,000 reaction rules are derived. These are used to train the neural network and combined with Monte Carlo tree search. Through the unique combination of this technology and data, we are able to deliver fast, robust, and innovative route prediction. Now let's discuss design and ease of use. Reaxys predictive retrosynthesis is fully integrated in Reaxys. Users are able to view published and predicted route in a single view. Commercially available starting materials needed for synthesis can be easily ordered using the shopping cart feature. The number of steps required in a synthesis route for easy comparison are provided. And a route topology is given to quickly assess the linearity or branching or complexity of the route. Further to this, in detailed tree view, the route topology is provided on the top for easily toggling between routes. Literature references of published routes that inform predicted routes, including the experimental procedures, are provided so users can make informed decision about whether this transformation will work. And secondly, what potential experimental conditions they can use to carry out such transformation. We have newly introduced the ability to extend route by adding one reaction step at a time. These reaction steps could be published or predicted, so users have more control now. And when you're happy with the route, these can be exported in multiple different formats for sharing or adding to electronic lab notebooks. We think we have built a predictive retrosynthesis solution on these three pillars, and we think it's a good solution. Let's now see how it performs in the hands of chemists. Over to you, Moritz.
Thank you very much. Yes, when we got first approached by Reaxys, we were delighted to be given the opportunity to work on such new groundbreaking technology as AI-driven or generally predictive synthesis starts to get bigger and bigger. We obviously were happy to be involved there. And so in order to get the most out of it and to find out the best data set for this program, we went out and divided our group into 7 teams consisting to 3 to 4 researchers each, and they were made up of senior and junior students within our group. So we have a large variety of different experiences within those smaller groups. These groups then selected 30 molecules from a variety of different fields, get a large structural diversity here. So as you can see down at the examples of some of the molecules chosen, we have very drug-like molecules, but we have also molecules that might very well be natural products or at least intermediates towards the synthesis of a very complex natural product. Groups then got assigned a couple of those molecules, and within the group came up with the first routes without using the predictive retrosynthesis tool. These routes were then carried towards other groups and were assessed for their scientific viability, while also factoring in how much time and effort was required in order to get to a synthesis which we were comfortable with. We then moved towards implementing retrosynthesis prediction into our routes and fed the molecules into the tool. At this point then, these routes were taken up by the groups and again compared with our generated routes, and also checked for their viability to find out which steps are likely to work and which steps may be problematic. Since the tool, however, keeps developing throughout the time that we used it, we sometimes went back a couple of times and actually fed a couple molecules, not all of them, into the tool again when new features were added in order to find out how these features improve upon the workflow which we can have for these molecules. So in order to see how the group actually rated the tool and how they worked with it, we want to quickly show the results of an internal survey we did. People had to answer a couple of questions. We can see that it was rated very highly on how intuitive it is to use and how user-friendly the interface is. Generally, the people here seem to be in agreement that for people who are familiar with Reaxys, they can translate that knowledge very well to this tool. Next, people were also very fond of the ability to review reaction conditions and literature references for predicted and published steps. The integration here was generally seen as very positive as well. A more mixed picture we see for the question of if the generation of new ideas is facilitated by the tool. And we see here that some people are very happy with it, while some people are not that happy, which we most likely attribute that to the fact that drug-like molecules are very well supported by the program, while more complex molecules often pose a bit of a challenge to the program and seems to stutter there from time to time. So without further ado, let's now move on to a couple of examples of routes here. First, I want to start with a more drug-like molecule, which looks rather similar to molecules that can be found in the development process of pharmaceutical companies. So firstly, we see the human-generated route, which chooses available cheap starting material from catalog retailer Sigma Aldrich, and then builds up linearly to the final product. The key step here being most likely the double reductive amination in order to form the bridged sort of pyridine ring. Moving on to our retrosynthesis tool route, we see that the approach actually is quite a bit different. There's not a more linear route chosen, but actually a convergent approach, bringing two fragments together in a last step in order to form the product. And again, we see that the step here is also formed, ring here is also formed through the formation of an iminium, which then does sort of a pinkish-Spengler reaction in order to fuse it to the imidazole ring. So as a takeaway for this, we can say that both routes are interesting and robust routes, both by our group, but also by the retrosynthesis tool. And we can say the conditions shown here are generally, especially for the retrosynthesis tool, are extracted by the groups who have worked on those molecules from the retrosynthesis tool. As always, multiple conditions are given by the tool. This again shows how easy it was for the groups to obtain quite good conditions for these transformations in which they had high belief that they will work. But furthermore, we can see that the tool and our approach differ in one key factor, and that is the choosing of starting materials. While we went back and went for more the catalog approach in order to find chemicals which we can easily access, the tool actually went to more complex starting materials, which can either be prepared over a couple of steps or potentially in an industrial setting, for example, be outsourced to a contract synthesis company. So we kind of already see here that the tool can provide insight in maybe very complex starting materials which are still available. Moving on to our second drug-like molecule, this was a molecule that was part of a bigger peptide project and was synthesized in our group. So we can see here the approach of our group is relatively straightforward. Key to the synthesis most likely being the Michael addition of the ethiolate into an enoate and trapping of the resulting enolate with decanal in order to rapidly construct the carbon skeleton here. Looking at the retrosynthesis tool route, we see a quite different approach. The starting material is still widely available in just cysteine, which is then transformed to the thioether. So here, actually, the first bond to form the 7-membered ring is the thioether, not the amide bond. We then see to close the 7-membered ring an amide bond formation, where we can see that the program might have a small hiccup here. As we can see, it tries to form a acid chloride in the presence of later aqueous HCl. This might not be the most feasible step, however, can easily be avoided by changing the order of steps and having the Bogdy protection in the first place. So we can clearly see that both routes here are robust, but the input of chemists is still very much needed in order to teach the tool some things that it might not know inherently, like some functional group stabilities or how they just perform in certain steps. But again, I would say a very, very solid route that was developed here. So if we move on and now look more at like natural product-like molecules, which are clearly a little bit more complex, we want to have examples of that as well. So looking at this is a molecule that was again part of a synthesis in our group and a fragment of a larger molecule. And we can see here that this decalin system is built up pretty rapidly in our route by a Konya-ene reaction, and then later through sort of reductive amination and coupling the carbamate is formed. Looking at the predicted route here from the retrosynthesis tool, we see an approach that, while sharing some similarities, actually is still a bit different. So through hydrocirculation and 1,4 addition, we form the precursor, which then under Claisen conditions closes the decalin here. However, it has to be noted, late game of the synthesis from the tool looked again very solid. One point that has to be highlighted though is the Wittig olefination of the diketone. It looks like this might be potentially a feasible reaction, as this ketone is most likely the more accessible of the two, but this is not completely safe. However, having the integration here of the retrosynthesis tool into Reaxys and so being able to quickly look this up through a Reaxys search, this can actually be verified or found out to be a problematic step, and then in order to potentially be replaced by a different step. So the integration here is a very important takeaway in order for chemists to have a deep dive into certain steps where they might not be completely sure whether they will work as predicted. Moving on to the final molecule, which again, it's sort of a derivative of a molecule that has been made in our group before. And the tool was chosen very deliberately because looking at this molecule, it has a very, for us, very clear disconnection within it. This is a nitrile oxide cycloaddition. So looking at this molecule, our route basically focused on that immediately, just as quickly as possible, generating the precursor for that, and then finishing up the synthesis of this molecule by nitrile oxide cycloaddition. We were kind of interested how the program would handle this, and to our surprise, we can say the program did not want to do a nitrile oxide cycloaddition, but do something completely different. In that case, it's an oxidative cyclization, which I have to state I wasn't aware of before the program told me that this is an option to do here. So this was very, very interesting because here this clearly we were very set out in order to find just the one route which we could think of, and the program actually provided something different. But, and if we look at the initial route, we see that now the program actually has a lot of problems. So we see the transformations shown here are as predicted by the program, and these are clearly not feasible. However, as a new feature has been added after some time working with the program, which is going through guided routes and actually doing it step by step through the tree search, we were actually able to guide the program towards a route which we think is pretty solid towards the cyclization precursor, and then could lead to implementation of this innovative step. So I would say this is actually pretty cool that this program sort of can eliminate a bias here that we might have had for this molecule, and actually give us something new here. And then even if it has problems at a point, we can actually use the ability to custom build a route, guide it towards a route which we are more comfortable with and think can potentially succeed. So what is the takeaway for working with this molecule, with this tool for us? What we can definitely say, the tool is very, very user-friendly. It is easy to use, and if you're used to Reaxys, you can very well translate your knowledge there to this tool. The routes it comes up with are also pretty robust, especially for drug-like molecules. However, they might still need some review at some points for steps which are not completely clear whether they will work. However, as the tool is very well integrated in Reaxys, reviewing these steps is very easy and brings us to our next point, that it saves a lot of time by having everything in one place. So being able to design a route, customize it, look up literature in one place makes this very, very time effective way of planning a synthesis. It has also to be stated that, as we saw in the last example here, these connections are pretty innovative at some point and can actually help to eliminate a bit of bias that we potentially in synthesis can experience at one point, because we're just so focused on one particular disconnection. So this is very, very cool, even for more complex molecules. These, however, are also the challenges. It's clearly for complex natural product that the program has a lot of issues, can often not provide a full route. However, with its sometimes very cool disconnections and guidance, it can actually be made to form pretty robust routes for these molecules as well. So where does this bring us? It brings us to the point that this tool is the perfect tool for a chemist to apply his knowledge or her knowledge on a synthesis. So this tool is not made to replace a chemist. It's very much a tool that adds to our toolbox and to our workflow when we want to create a synthesis plan for a molecule, as we can basically use this in order to apply our knowledge and get more and more information out to make such molecule. So I think this was a very important takeaway for us. This program is a lot of help to us, and it's very, very good for our workflow. It really also still needs our input in order to provide very, very nice routes. So this interaction between those two sides is very important. And we can see this with the feedback that the group gave. A lot of people were quite happy about how the program made them able to access literatures about the disconnections that they wouldn't have thought of, and especially also how much time it saved for these like drug-like molecules where the provided syntheses are very, very good to start with. So, however, working in this collaboration is also fantastic because we can actually give feedback for things where we think there is more that can potentially be added. And this is so great about it. We're users here, but we can really give input as well. And there are some things which we would like to see in the future potentially to be added. A lot of people were quite in favor to get even more potential to guide this tool, having options to protect certain bonds or to clearly state, I want you to break this bond in order to help the tool even more for these more complex molecules, for example. Some people wanted to have be able to define certain intermediates or starting materials. This might be interesting. For example, if somebody wants to do a molecule starting from chiral pool, where there's just a limited amount of molecules available. And last but not least, it is also a wish from a lot of people that they are able to exclude certain steps from the predicted syntheses. These might be to the fact that we're not able to run those reactions if there are very specific reactions where we are lacking the equipment to do the reactions. Also, it might potentially be because we believe that this step, the tool predicted at that point, cannot work in the setting. So being able to exclude that step might help us also to develop more complex routes for complex targets, which we really believe in. With that being said, it was a pleasure for me talking about this, and I want to give it back for the future of this program to Abhinav.
Thank you, Moritz, for that tour de force. And with that, let me just show you. So, besides working with Moritz and ET at Zurich, we also work with a lot of pharma and agrochemical companies, and predominantly the feedback that we receive really align well with what you have heard from Moritz. So the tool that we have built is user-friendly and very user-friendly. It does provide innovative disconnections and helps with and saves time with synthesis planning. And really the routes proposed for especially drug-like molecules are mostly robust. We are constantly developing the tool further, and feedback we are, our roadmap of development is user-centric and guided by chemist feedback. So we are very, in a way, very thankful to Moritz and Professor Carrera's group for providing valuable feedback. As you heard from Moritz, so chemists want more control. So that's what we are working on. So to give our roadmap is divided into 3 parts. Giving chemists more control. So to be able to define bonds to break, to be able to define intermediates to be included or excluded. Second, to improve and augment the predictive model even more. What this means is that, firstly, we update the model with the latest reaccess data because every half a year, you have a lot of new chemistry being published. Every quarter, there's new chemistry being published. Second, to improve the diversity and of the routes that are proposed, as well as improve the respecting of functional groups, as Moritz was alluding to earlier, and regioselectivity. And importantly, what we also are working on is to give the ability to customize the model by including or by training the algorithm on combined reaccess and customer reactions data. Because a number of companies, some academic groups use electronic lab notebooks, and there's a lot of reactions data sitting in there, which will be, which can be combined to make the algorithm more stronger. Lastly, we are working on improving the ability to edit synthesis plans even more. So giving the users the ability to delete steps that they do not like, delete routes or projects that they do not, delete routes that they do not like. And importantly, which would be delightful for all the chemists, that to be able to export it as the retrosynthesis tree as a ChemDraw file. So that, it makes the sharing even easier. With that, I would like to, I hope we have been able to show that, the tool is built on these 3 pillars and really augments chemist knowledge and helps accelerate synthesis planning by providing robust route suggestions. I would like to thank my collaborators at ETH Zurich, Moritz and Professor Carrera's group, and also the whole Reaccess team. So it has been a great working experience with Moritz, and we continue working. It's not, our collaboration runs further.
Thank you, back to you as well. It is a pleasure. It's truly fantastic to work with such a great team on such a great field that is developing that rapidly and being able to have that insight into this and also learning on how to use it for our own workflow has been fantastic.
Thank you very much, everyone, for listening to us.
Thank you.
예, 감사합니다.
Thank you very much. That was the last lecture. The last lecture was about how AI is used to accelerate synthesis. And AI and big data, the new world of innovating biohealthcare was the theme of this session. And one thing I would like to add is when we use anti-cancer drugs, they don't work on all the patients because they have different effects. When the compound A is used on a cancer cell, sometimes it could be effective, and I don't think we've heard much about omics data. Whether we do the genetics analysis, we shouldn't just target the cancer cells. We have to also look at the TMs because they undergo mutations. We have all these genetics data, and we have to have a pool of various data to be able to solve the problem. Chemistry, big data, personal information, biology, and personal biological medical traits. they have to be pulled into data to be able to accelerate the value. And this may only fall as an empty dream. In order to enhance personal health, we need the technology and the industry, but we need to have data convergence and recreation of the value. And I'd like to thank everyone for joining us at this morning session. We will be back at 1 p.m. with the sponsor session by Hanmi Pharma. We look forward to your participation. Thank you very much.